So as we move into chapter 4, we're somewhat getting away from this whole chemistry, atoms, protons, all this stuff, and getting into what cells do. I know that you're longing for the day that we get to the human body, because this is human A and P. Are y'all moving into systems in lab yet? Y'all finished the histology part? Okay. So that's when it gets fun. This chapter is the last foundational chapter that the A&P folks want you to have some experience with. The nursing folks want you to have some experience with. So we've got this one more chapter to go about cells, and then we'll move into the integumentary system before we get to our second test. Now, I also wanted to point out, which I didn't, on our calendar, if you notice, I put these chapters in green. This is the information that you're going to be responsible for the exam 2. But notice there's some red. This is going to be the chapter on, I believe, skeletal system. We may begin some of that material before we take the test, but that will not be on the second test. I just didn't want to finish covering our material here and then have a test on a Wednesday. I like to give you a weekend from when you get that last material to the second test. So just pay attention to those colors.
Which chapter 5 are?
I don't think we're covering chapter 5.
Okay.
Yeah. All right, so cells. There are a ton of cells in your body. I mean, there's a ridiculous number of different kinds of cells, different shapes of cells. But the one thing we can say about cells is they're all pretty small. And what can we say as far as a cell relative to the definition of life? Remember that one from? Smallest unit of life. Anything smaller than a cell can't be alive. It can consist of molecules and be put together and help cells, but it's not living by our definition. But cells can be small, but cells don't have to necessarily be small. I mean, we've got bacteria in the neighborhood of 0.3 of a micron. Our average animal cell is 10 to 100 microns. But look at a nerve cell. Man, we can have nerve cells that are longer than 1 meter. Now, again, they're small in diameter, these processes that extend that far. But they're still small. In fact, when you look at your red blood cell, that's going to account for approximately in this neighborhood of 10 microns. Your red blood cells are actually 8 microns in diameter, but I like to stick with numbers with zeros and 5s. Just kind of easier to remember that way. So red blood cells are around that 10 micron in diameter. So we have to stop and say, all right, we have all of these teeny tiny cells in our body. Why don't we have fewer cells and just make them larger? I mean, you'd think that might save space, save energy. Well, when we look at biological systems, and I know we've talked about this before with the inner folded membrane of the mitochondria, we're always focused on surface area. And in this case with cells, by having many smaller cells, then what we have increased is basically the surface area of our plasma membranes to the volume that's inside the cell. Because if we took a ginormous cell, it's got a humongous volume, but not very much as far as a surface area. And that is really going to affect this rate of diffusion. Now, we talked about diffusion a little bit before. We're going to spend some more time with diffusion and other methods of transport here. But diffusion can only happen to a certain distance. And beyond that distance, molecules will not be able to effectively, in a timely manner, diffuse from point A to point B if it's too far away. And our plasma membrane is going to be that selectively permeable membrane, and we'll expand on what that means, through which diffusion can occur. But again, by having smaller cells, we have a greater capacity to maintain local concentrations of solutes, of substances, and we also have a greater capacity to diversify function. We're going to have some cells in the body that their role is to eat and destroy garbage and pathogens. Well, these cells are going to be right next to the cells that are allowing oxygen and carbon dioxide to diffuse in and out of your blood. If it was just one big cell, they couldn't really accomplish both at the same time. So, surface area to volume, this is one picture that helps me really get a perspective on what's going on. We've talked about our shape of our red blood cells. What is our shape of a red blood cell? Okay, that's the scientific term, biconcave disc. What is our term? Squished jelly donut, right? And we did that because we couldn't increase surface area. Red blood cells have to be small. Can anybody help me out? Why do red blood cells have to be that 8 microns? Why do they have to be that small and not larger? They have to fit through the capillaries. Capillaries are the smallest vessels in your body. Those are the vessels and the only vessels that allow the exchange of materials. Go ahead and write that down. That's going to be important. You will get that all through your A&P 1 and 2 and so on. Capillaries are the smallest vessels that allow the exchange of materials. Gases, nutrients, wastes. So if that's the smallest diameter vessel, these things have to fit. And they fit in such a way that they go single file through the capillary. They look like stacks of pennies. Do what? Capillaries. Your capillaries. So our red blood cells, we can't increase diameter. Yes? Capillaries are the smallest, are the only vessels that allow for exchange. I mean, that's a little short nugget, a little sound bite. But if you remember that, it will help serve you in all of your medical training and education. Capillaries allow exchange, no other vessels. So we can't increase the diameter, but we can't have such a large surface area to volume because we have to be able to diffuse oxygen in and out. So how can we increase our surface area to volume if we can't change surface area? Well, we squish the middle. You see how they did that? So you can either fold up the membrane and have more membrane, or in this case, get rid of some of the volume by squishing down. Now, our intestines are another place where we have a lot of diffusion. We call it absorption. Absorption of a lot of the raw material that you ingest. And so the way we increase surface area to volume, again, unlike, well, almost the opposite of the red blood cells, we fold the membrane up again and again and again. And when you get to the digestive system and you get to your intestines, you're going to see that you've increased surface area to volume in your intestines because your intestines are 30 feet long. In addition to that, on the inside of your intestines, the inside layer that's doing the absorption, it has these folds that project out into the lumen of the intestine, maybe 3 of them. It looks like those fins in your dryer on the side that help kind of mix up the clothes. That's the same thing. They're called plicae circularis. It increases surface area. But then again, all the cells that are on the inside layer, they're folded up into these things called villi. That increases surface. Well, that's not enough because on the apical surface of all the cells, you have all these little folds of the plasma membrane called microvilli that increase surface area. That's for the digestive system. You don't have to write all that down. That's not going to be on the test. Are you erasing it all now? No, I'm kidding. But the point is, in your intestine, there are 4 different levels in which the intestines have increased surface area by having more membrane with which to absorb. And that's going to aid in this process of absorption, which largely is going to be mediated by diffusion. So in light of that, and having to get to what we are diffusing across, which is going to be our plasma membrane, this is where we're going to begin to see what the cell's doing and how material is moving in and out of a typical animal cell. Here we have an illustration. On the left, this is a transmission electron micrographic view of a cell. I'm not going to ask you to label any pictures on the exam, so don't fret about that. But we can see our yellow or orange plasma membrane. We have our nucleus, which is in purple with a lot of holes in it. Did you know your nucleus wasn't solid? It has these pores in it because material has to pass in and out of the nucleus. And then we have our Golgi here in purple, our endoplasmic reticulum in blue, and we've got our orange mitochondria that we've already talked about quite a bit, and we'll just touch on it and review it just a little bit more as we get to our organelles, probably hit those on Friday. But first, we need to talk a little bit more about this membrane, the plasma membrane. The plasma membrane is going to protect and support the cell. It's going to protect the cell, meaning that there are materials that are part of your immune system that are going to be tasked with identifying pathogens, identifying foreign material. And your membrane is going to help protect the cell from many of those immune factors. The plasma membrane is also going to assist with cellular communication. Now, a lot of this is going to be facilitated through proteins, and we're going to talk about that more as well. But sometimes the communication between cells is direct contact. When you study about smooth muscle, you're going to see there's some direct contact. Cardiac muscle, cells are directly in contact. We will, at the end of this semester, talk about the nervous system, and that communication is going to be sent through chemicals. One neuron is going to secrete chemicals called neurotransmitters. Another neuron is going to detect those signals and interpret that. The secretion out of a membrane and the detection of the signal through the membrane, that's all part of our communication. And in some ways, like we said with secretion, that's going to be regulation of transport. And so the membrane, it's not just a barrier. And I don't want you to think of the plasma membrane as a hard plastic bubble or like a basketball that is completely solid in which air can't get in and air can't get out. We're going to see that the membrane is actually pretty fluid. So the plasma membrane, in addition to the support, it defines the boundary. The boundary of a cell that's meant for contraction versus the boundary of a cell that's meant to send a signal versus a boundary of a cell that's cleaning up debris. It separates the cell and it defines certain functional zones and distinguishes them from a neighbor. Now again, cells and the plasma membrane, it's not a concrete layer. And when we look at our membrane, do we remember what molecules make up our membrane? Phospholipids. And what category based on charge and such? What category of molecules are phospholipids based on charge? There's that one special term, remember? Amphipathic. They're different on the two ends because one end, where we see the little orange circles, that's what group? The other one. Hydrophilic. The polar group, the polar head, versus our squiggly tails. Those are the, now you got it. Take the floor. Hydrophobic. Good answer. The tails, the hydrocarbon chains, those fatty acids, are hydrophobic. So we have our phospholipid bilayer. But what are they illustrating here with this big purple spherical alpha helical thing right there? What is that purple thing in our phospholipids? Alpha helix is your clue. It's not a beta sheet. That's an alpha helix. Proteins. There are proteins that are embedded or anchored to or span through the plasma membrane. Now, with the lipids, we're going to see we have cholesterol. We're also going to have carbohydrates attached, but especially with these proteins. The understanding that the plasma membrane is not a concrete, immovable object, it's sort of stated in this name called the fluid mosaic model. Fluid means moving. And mosaic, have you ever seen a mosaic painting? It's got all the dots, and if you're looking really close, you can't see what the picture is, but if you move far enough away, now you can see this one image that's made up of all these little dots. And so in this idea of mosaic, meaning the membrane is a lot of different components. Phospholipids, carbohydrates, cholesterols, proteins. So putting those two concepts together, they come up with fluid mosaic model. But I want you to remember, just like we had on the previous test, the primary component of a plasma membrane are the phospholipids. And in fact, when you look at the number of phospholipids to the number of proteins, it's a 50-to-1 ratio. There are many more phospholipids than the proteins. But this is the part that's kind of sometimes difficult to wrap your brain around. Even though there's so many more phospholipids, the phospholipids are very, very small compared to the proteins. So while they're 50-to-1 phospholipids to proteins, when you look at just the mass, the amount of stuff that makes up the membrane, it's 50-50. Half of the membrane is phospholipid, the other half is protein. It's just from a weight standpoint. Now, I've tried to embed a little video clip that really gets at this fluid mosaic model. I think it has sound. I'm not sure how it's going to look or work, because the last time I tried this, it was real slow. Because as soon as I hit this button, I think it's going to start. So I'm kind of holding my breath. Let's see what happens.
The cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
But it was cool because it showed these proteins allowing molecules to go through these protein channels, and it showed how the protein, like a big ship on the ocean, was just kind of floating around. That's the whole idea of the fluid mosaics. And in fact, those phospholipids, they can just sit there and spin around, and sometimes they can flip from one layer to the next layer. So each phospholipid's not glued in place. It's just those molecular interactions, primarily because the polar heads can attract water and the hydrocarbon tails don't. So they just sort of accumulate together. So most of our membrane, most of our lipids, and the principal lipid are the phospholipids. Then we also have the cholesterols. The cholesterol, remember, the part that stabilizes the membrane. Cholesterol acts as a fluidity buffer. It doesn't allow it to get too fluid when it warms up, and it doesn't allow it to get too solid when it cools down. But then we also have these things called glycolipids. What does glyco mean as a prefix? Sugar. So you see we have these hydrocarbon tails, fatty acid chains that are attached directly to the carbohydrate. So the fatty acid tails insert into the middle of our layer, and then the carbohydrate sticks toward the outside of the cell. And in many instances, these serve as markers for the cell to say, hey, I'm on your team, don't destroy me, I'm the good guy. And in fact, carbohydrates on proteins, glycoproteins on the surface of your cells, play a very important role in your physiology. So if you were going to the hospital to have a surgery, and there was a chance that you were going to lose a lot of blood, what's one very important piece of information you would want the hospital to have? Your blood type. So when you study blood, and again, this is probably going to be an A&P 239, I want you to remember that the protein, this protein marker that's on the surface of your red blood cells, it has carbohydrates on it. And if it has the standard number of carbohydrates on that protein, which is called the H antigen, don't write that down, this is a freebie, then you have O-type blood. How many O-type folks do we have? All right, you have the H antigen with a standard array of carbohydrates. How many A-type blood folks we have in here? All right, so you have the same H antigen as our O-type students. The only difference is your H antigen has one additional sugar molecule. That converts the H antigen into the A antigen. How many people have B? You have the H antigen, but instead of the sugar that went on to make it A, you have a different sugar that goes on to make it B. So you see, carbohydrates are really telling your body, okay, I'm A-type blood, I'm B-type blood. And if you have AB blood, some of your markers have the A sugar on them, and other of your H antigens have the B. They're not going to have both those sugars at the same time. So do you see how the sugars are important for the identification? Now, we've got our phospholipid bilayer. And while it is flexible, it is still pretty strong. And so there's some molecules that are not going to be able to pass through. They're not going to be able to diffuse easily. But when we look at the molecules that can make it through, they're going to be small and they're going to be nonpolar. So when we look at small nonpolar molecules, first one that comes to my mind is oxygen, carbon dioxide. And unfortunately, one molecule that doesn't really go by this rule, but that can still diffuse through the plasma membrane, is water. Because remember, we've already played that game with our hypertonic, hypotonic, isotonic. So water can diffuse through the membrane, not incredibly quickly. And when we look at our kidneys, we have a special water channel. And these water channels in your kidney tubules are called aquaporins. Again, you'll get to 239. But that speeds up the rate of diffusion of water. What is our term for the diffusion of water? Osmosis. So, since really only small molecules can pass unassisted, that's one of the reasons why we have all those proteins. We have a lot of protein transporters. And again, even some that can transport and allow water to diffuse through them. We have enzymes on the plasma membrane, not just inside the cell. And in fact, back in the kidneys, we have enzymes that take sodium bicarbonate and convert it into CO2 because CO2 can diffuse into your cells. And once into your cells, it takes the CO2, converts it back into bicarbonate, and puts it back in your blood. That's how your body reabsorbs any bicarbonate that maybe made it into the filtrate in your kidney. Now, cells have to be able to anchor. You don't want your cells just floating around in your body. If so, we'd just be a big pile of goo. And then we've already mentioned our receptors and our identity tags that we have on our cells that just, lets our immune system and especially know we're on your team. We're the good guy. But cell anchorage is important, cell-cell adhesion. Without cell-cell adhesion or cells adhering to their underlying connective tissue, your skin would completely slide off. The bottom layer of your cells, the reproductive layer of your cells that are making more and more like a conveyor belt pushing up, those cells are attached to molecules in what is referred to as the basement membrane. It's the beginning of the connective tissue. And one of those molecules sitting out there is a molecule called laminin. And if there's something wrong with laminin or the receptor on the cell that hangs on to laminin, your skin will start forming these huge, horribly painful blisters. And that disease is called bullous pemphigus. You don't have to write that down, but hopefully you can understand how anchorage is absolutely essential. All of these functions of our plasma membrane proteins are absolutely required for homeostasis and proper health in the human body. All right, so we've talked about diffusion a little bit, talked about osmosis a little bit. You're not surprised, right, that cells need a constant supply of water? Why would you think they need a constant supply of water? That's what we're mostly made up of. And we're continually losing water vapor through our skin, through our breath, so we continually have to bring water in. We're continually losing water from our urine. You can be in the hottest desert on the planet, but your kidneys have to eliminate 300 to 400 milliliters of fluid a day to remove the nitrogenous waste from your body. That's called obligatory water loss. Again, that's a freebie. You'll get that in 239. But water is essential. Food, yeah, that's a no-brainer, right? You've got to have this raw material to supply energy. But again, those wastes that I just mentioned, we have to eliminate the nitrogenous waste, the breakdown of proteins. CO2 is a byproduct of what process? What process would you say CO2 is a toxic byproduct of? Respiration, and specifically cellular respiration. We have to get rid of it. And then, of course, the extra water. We can't have too much water. Overhydrated is just as bad as dehydrated. In fact, you can die from too much water, right? Have you heard about those people that if you keep your hand on the car and you don't go to the bathroom for a certain length of time, or who can drink the most water can get a car? Yeah, too much water will kill you. That's why I always say too much of anything is too much. All right? Too much oxygen. You know too much oxygen is a bad thing? Yeah. Too much oxygen, too much water, a bad thing. So our cells have to maintain all of these materials at the proper concentrations and transport them appropriately in and out of the cell. So understanding the plasma membrane, understanding the components of the plasma membrane, and then understanding how cells transport these materials are absolutely essential to understanding how cells can stay alive. So again, here's an illustration of our cell. This is showing transport happening not just at the plasma membrane, but across the membranes of all of our organelles. But these are the key points that we need to absolutely remember. Selectively permeable, that's our membrane. What does selectively permeable mean? Let's just make it simple. Okay, let's make it even more. What's permeable mean? Yeah, something can pass through it. Permeable, something can penetrate through the membrane. Selectively, what does that mean? not everything. Sometimes it depends on what. So some things can pass through the membrane. Now, selective transport means that materials that can't simply diffuse in and out of the cell on their own have to be transported. Well, not every transport protein is going to transport everything. And in fact, when we look at the nervous system, we're going to have a specific channel that transports sodium ions, and we're going to have a separate protein channel that transports potassium ions. And that's all they transport, period. So these transporters are going to be selective. Now, once again, what passes? Ions. How do ions get across? Yeah, they're charged, but we said nonpolar stuff passes. So how do ions kind of get around that? They're small. Some small ions that are uncharged, they can get across. Sodium and potassium won't. That's why we have to have those transporters for those. Organic molecules. What's another way we can define organic molecules? Nonpolar molecules, hydrophobic. Hydrophobic molecules easily diffuse in and out. Now, it says some macromolecules, but most macromolecules, and I'm counting glucose as a macromolecule, those have to have specific transporters to get them in and out of a cell. Yes, ma'am.
You said that selective transport and that one's transport as well?
Selectively permeable just simply means permeable means things can pass through the membrane, the phospholipids. The transport is going to have to use a protein to get through. It won't get through the membrane on its own. All right? So as we look at transport, again, we've already talked about diffusion a little bit, especially as it relates to water and osmosis. So we're going to review that just a little bit, but then we're going to talk about facilitated diffusion. Now, facilitated diffusion is going to be a transport. It has to use a protein. And then we're going to compare diffusion to active transport, and as the name implies, you're going to have energy for this. So on Friday, we will start looking at these different divisions of transport across the plasma membrane.